Okay. Restoration strategies have become more critical over the past decades to assist in the recruitment of corals. One strategy involves culturing sexual coral recruits from gametes to improve the genetic diversity of planted corals. However, one of significant bottleneck challenges in this process is the sediment, the post-sediment survival, and the coral recruit growth, mainly due to competition with fleshy algae and predation. Ex-situ sediment can be enhanced by using substrates containing specific characteristics to attract larvae, and the post-sediment survival might also be enhanced by using substrates with sulfur properties that may promote long-term larvae addition. The objective of this project is evaluate the synergistic effects of specific physico-chemical properties of substrate coatings in relation to sediment patterns and behavior, as well as post-sediment survival for Acropora palmata, Orbicella annularis, and Orbicella fiolata. The first experiment is coatings with combination properties, and the methods are the substrate fabrication. We prepare 6 coatings. The substrate is IDs, the C1 to C6, and everyone has intermediate hydrophobicity polyester design except the coating C6. Then we put micro or nanoparticles with with phosphorescence with different colors. The C1 has light blue, the C2 a red orange, and C3 turquoise. And the substrate C4 had a hydroxyapatite, and the last ones, the C5 and C6, had a calcium carbonate. The substrate C2 to C3 have a superhydrophobic polysiloxane coating, and the C6 have this coating. We prepare 20 replicates for each treatment. The experimental design. For Acropora palmata, the substrates were conditioned. 10 replicates of each treatment were placed in fluid-through seawater aquaria for 4 weeks to allow a biofilm formation, and 2 experimental designs were carried out. For the nutrient experiments, each plastic container was fitted with 1 conditioned and 1 unconditioned substrate with the same coating. The experiment consisted of 5 replicates of each of the 6 coatings. And for the Sherrick's experiment, each plastic container had 2 substrates of each type of coating, one of which was conditioned, and whereas the other remained unconditioned. The same experimental design was repeated for Orbicella annularis and Orbicella fiolata, but used to unconditioned substrates. And then we transferred the primary polyps to the fluid-through aquarium. And the variables that we evaluate here is the sediment between the 6 coatings, the sediment between conditioned and unconditioned substrates, and the survival between the coatings. And the statistical analysis where a generalized linear model was performed to determine the effects of substrate coatings and substrate conditioning. And a Kaplan-Meier survival analysis was performed to determine differences in survivorship within substrates. Okay, the results. In the 2 Acropora palmata experiments, no significant differences in the sediment were found between the 6 treatments. But for Orbicella annularis, we found differences between the substrate C1 and C4 and C6. The sediment was higher in this substrate, and this substrate had a high hydrophobicity and a light blue phosphorescence. This substrate the sediment was higher in the substrate compared to substrate C4, and this had high hydrophobicity with red-orange. And we saw the same result with Orbicella fiolata, was higher in the substrate compared to C4. And these results can mean that this species can use photosensitivity, and these respond to color, and in this case light blue and red orange, to fine-tune the selection of appropriate substrates or microhabitats. The conditioning. No significant differences were found between the conditioned and unconditioned substrates, and it should also be noted that none of the substrates used in the Orbicella annularis and Orbicella fibrillata were conditioned. Hence, no biofilm could have formed at these surfaces. Yet, larvae settled, which could mean that conditioning is unnecessary for these substrate types. The survival. We saw that this graphic is for Orbicella annularis and the other one for Orbicella fibrillata, and this x-axis is time, days after spawning, and this is the survival probability. And for Orbicella annularis, the survival was higher compared in the substrate C5 compared to substrate C2, but for fibrillata we don't find differences. So, on transferring the recruits to arid aquaria, the number of polyps that survive on the substrate drops abruptly, especially those coated with polystyrene. This was evidenced by the fact that the longest survival time was associated with the only substrate without polystyrene coating, that is C6. So, this is the higher survival. C6 is the blue. The type of polystyrene used somewhat soluble in water, hence the resin may help slowly dissolve, resulting in an unstable material, possibly preventing viable long-term sediment and post-sediment survival. The hydrophobicity generates a weaker adhesion force between the recruit and the substrate. When causing movement in the aquarium system of aeration, the recruits detach from the substrate since they were possibly very weakly attached. Using these hydrophobic surfaces to prevent the colonization of other organisms, such as filamentous algae, can also be detrimental to coral survival since they work like antifouling coatings that are easy to release when there is movement. So we do other experiments because we want to evaluate the properties individually. So the objective of this project, this other experiment, is evaluate the individual properties of the substrates that elicitate the sediment preferences of Orbicella species to determine if the sediment preferences is determined by the combination of the properties or by some individual property. We focus on evaluating the properties of the substrate C1 and C2. We prepared 6 coatings. There was 4 treatments, and we implemented the controls because in the other experiment we did implement controls. So to the 4 treatments, there was T1 to T4. The substrate T1 has a super hydrophobic polysiloxane coating. The T2 has a micro or nanoparticle, the microphosphorescent light blue microparticles, and the T3 has the red-orange phosphorescent microparticles. The T4 has only the hydrophobic polyester resin. The T5 is a ceramic floor tile with white paint, and the T6 is only the ceramic floor. Okay, the experimental design is 10 substrates of each treatment and control were placed in flow-through seawater aquaria for 7 weeks to precondition them and allow a biofilm formation. Each plastic container had 6 substrates, the T1 to T6, but 5 plastic containers had conditioned substrates and the other 5 plastic containers had unconditioned because we want to evaluate this independently. And the post-sediment survival, the primary polyps were transferred to flow-through aquaria with access to natural light, food, and cyanides. The results. For Orbicella annularis, differences were found in the number of cells between the substrates. T6 that was higher compared to T2 and T4. T6 is a ceramic floor, is a control. And the T2 is with light blue phosphorescence, and T4 is with the resin coating. And for Orbicella fibrillata, we don't find significant differences between them. So The combination of properties is what generates a successful settlement on these substrates, but individual properties did not generate this effect. And the conditioning. We found differences between a substrate conditioning for Orbicella fabulata, but for Orbicella neuralis we don't found differences. We found a higher sediment in conditioned substrates compared to a condition, so this could be that the substrates with a single property are not attractive enough on their own to larvae and require conditioning to generate an adequate sediment signal for coral larvae. And the survival. No significant differences were found between the substrates for the any one species. So we saw that we have the same results of the other experiment. The longest survival time was in the substrates with a resined polyester and hydrophobic coating. And in conclusion, this is the first study to report the ability of Orbicella annularis and Orbicella fabulata to detect phosphorescence and hydrophobicity during the substrate selection. And this is the first time that the effects of substrate surface properties have been tested on these species with varying results. On the one hand, microparticles with blue and red-orange phosphorescence and high hydrophobicity favor sediment in Orbicella species, but some of the components may be detrimental to the survival of the polyps over time. These issues need to be addressed as long-term survival is critical in restoration projects involving the outplanting of coral recruits. Finally, it is essential to keep testing different combinations of substrate properties to determine the most conducive to larvae sediment and polyp survival. This will help us identify the most suitable substrates for each species, which will be vital for optimizing future restoration efforts. Thank you so much, and thank you for my partners of Corallium.
Thank you very much, Laura. That was perfect timing. That was beautiful. We have time for a couple of questions. Do we have questions from the floor?